Thanks, Betsy. And I don't know if it augurs well that RWJ Aspen whole shebang is over now.
Had a good run, though.
Yeah, we had a good run. And thank you, everybody, for coming out on a yucky day. And I have to say, talking to Ian just before, I'm probably much more nervous than I thought I'd be for this talk, considering everybody who's in my empirical seminar, who I've been hammering for 3/4 of a semester, now get their revenge on me. So today I want to talk about this scary title, The Bell Curve Revisited, and the outline is such that I'm going to give you this sort of motivation and background. Then I'm going to get into some details about how we measure genotype, and then I'll get to the empirical analysis, which will test 3 hypotheses that were put forward by Herrnstein and Murray back in 1994. And then I'm going to suggest an alternative, I think, more productive way to integrate genetics and social science moving forward as we leave the bell curve behind, and some of the social implications of the social genomics, what I call the social genomics revolution, going forward for society as a whole. So for me, my journey began not the bell curve in 1994, but a couple years later in 1997 when Susan Mayer wrote this book as I was finishing my doctoral dissertation, in which she used a number of clever tricks and brought the economics causal revolution into sociology a bit, where she showed that the traditional regression-based naive estimation of the effect of poverty, or really income more generally, on children's outcomes was vastly overstated due to unobserved heterogeneity among parents that drove both child outcomes and parental wages or income. And so 2 solutions to that is, one, to go find natural experiments that essentially result in randomized income differences among families and look at the kids, and lots of work has been done in that regard in the last 20 years. And in some ways, the effects of income have been a little bit restored. I mean, there's a lot of debate about how big the effects of income on children are. Another strategy that I want to talk about today is to actually just go straight into that unobserved heterogeneity and ask, what is it that we're not considering when we just naively run an OLS of, say, parents' income or wealth or any socioeconomic variable on kids' outcomes. And there's really, at least in stratification research, where we're talking about intergenerational transmission processes, there's really 2 possibilities. I put this question to Susan at one point at a conference very directly, and she kind of evaded it. But what she thinks the alternative story is, there's probably more, but for me, it comes down to the transmission of cultural capital, some sort of internal household familial culture that's passed on, and that is actually the causal agent for economics and cognitive success among children, but also predicts wages among adults. And that would seem, if we could actually measure it, to be policy malleable, but would probably require a different way of thinking. I mean, some of the experiments now that try to increase amount of words spoken at home after the Hart and Risley study and so forth are probably working on that model. The second confounding variable really is what economists would call endowment, what we might call sort of innate ability or something that's baked into our biology through the genome, and also, of course, is passed on from parents to kids. And that's the sort of more concerning or troubling or intriguing, if you will, alternative hypothesis that Herrnstein and Murray went straight into in arguing for the rise of cognitive stratification in the United States. So what do? They use the NLSY 79, and that's important to keep in mind, and they measured cognitive ability. Of course, we know, even with what's called the 4th law of behavioral genetics, that everything has a genetic component. That's Eric Turkheimer at the University of Virginia. If you just measure IQ or cognitive ability, that's got both genetic and environmental components. It's endogenous to all the things we're interested in. They argued that there was high and rising returns to IQ in their data. However, look how narrow their window, their birth cohort window is using the NLSY 79. They're really looking over a very specific time period, 7 years really, over the start of the third of the 20th century. So I'm going to go back and I'm going to look at birth cohorts in the HRS that were born from 1920 to 1960, which really is a much more representative sample for when we think the rise, when we associate the rise of higher education in the United States and the knowledge economy. It's a little before the knowledge economy, these folks, but this is the period of expansion of educational opportunity in the mid-20th century in the United States that we should see their effects. And I'm going to use genetic data. They were making a genetic argument 20 years before it was possible to actually measure the genome in any systematic way. By the way, feel free to interrupt me at any time. I'm a New Yorker, and I also prefer the economics approach of a more interactive presentation rather than the sociology. Hold it back like a dam for 45 minutes. Recent genotyping technology has fallen in price faster than Moore's law for microprocessors. Today, you can genotype I can take your saliva or a sample of your blood, and I can get your about 1.5 million markers, which can then be imputed to 3 million. And there's about 30 million sites of variation in the human genome. We get 1/10 of those through imputation for about less than $100. If I did all of us, I could probably do it for a bulk rate of like 120. I'm actually pricing this out right now. So, and most of these chips that are now cheap and easy to do focus on one type of genetic variant that's called a SNP, or single nucleotide polymorphism. As you can see, there's copy number repeat variation here, but most chips focus on this. And this is simply, if you remember your biology, there's 4 bases in DNA, and one person has a T there, and it's been substituted through mutation to an A in another part of the population. So, so early work focused on particular SNPs and failed to replicate, had a lot of problems because those SNPs weren't distributed randomly in populations, so they're confounded with environments and so forth. So new approaches that have been used more recently and have been expanding is to create a polygenic score, which is a very simple exercise. As you see in the HRS, we had almost 1.5 million SNPs of these sites of variation that are biallelic, that there's only 2 possible, A or T or C or G, not complicated, that where there's 4 different versions in the human population. And you run essentially 1.5 million regressions, very simple, with some very basic controls, and you get a beta for each SNP, and then you go to that's in a discovery sample. So this one that I was involved in involved a meta-analysis of many cohorts across the Western world, including UK Biobank, Swedish Registry, Twins UK. There's an enormous number. It was an enormous organizational effort by the Social Science and Genetics Consortium. And through that meta-analysis, we get these weights, and then we take it out of sample, in this case the HRS, the Health and Retirement Study, and we take every individual who has either 0, 1, or 2 of those reference alleles at that location, and we multiply 0, 1, or 2 times the beta, and then just do that 1.5 million times and sum it. Very simple. I mean, there's more complexity buried under there about the fact that the SNPs are not sorting independently and so forth, but basically, especially for the social behavioral outcomes that I'm going to talk about, this just simple, raw, throw it all in approach has seemed to be the best. So when we do that, we get an R-squared of almost 6% in the HRS for education. So this is the second iteration of this exercise. And it's really a matter of sample size. We expect that to rise to double digits in our next paper and eventually level out at about 20%. We know that from other analysis of what the ceiling should be. So let's just pause for a moment, because I think it's a pretty amazing thing that I can take your saliva, or I can take a cohort of infant saliva, and I can explain 6% of the variation in schooling 25 years later, or 30 years later, based on that saliva, under very few assumptions right now. So I mentioned that we got 7% approximately. This is in a different out of sample, but 7% R squared for education from this polygenic score. We expect the max to be 22% based on another approach called SNP-based heritability. These keep changing as new analyses come online and sample sizes go up. So you can see that for height, we're up to 17%. A single variable will predict 17% of the variation in population height in white non-Hispanic populations. So what does this PGS, or polygenic score, do, or what does it mean? As I mentioned, Herrnstein and Murray and other previous scholars have used measured downstream endophenotypes to education. In other words, they measure cognitive ability. That's most typically, and sometimes other studies will also throw in some non-cognitive measures. But of course, the problem is that those are downstream and endogenous, as I mentioned. And a whole other strand of research has focused on twin or sibling models to estimate the genetic and shared environmental components to outcomes like education. However, those by definition are latent variables, and then therefore not very useful to us as scholars who want to do something else with the genetic component. We want to play around with it, as I'll talk about. We want to use it as a control variable. We want to look for gene-environment interactions and so forth. You can't really do that very well with a latent twin model. So the PGS in the Nature paper, we also looked at gene expression data, and we found that the top hits, the top 74 genome-wide significant hits, were associated with central nervous system endophenotypes. However, I want to emphasize that this is a measure that encompasses everything and anything. So to the extent that there's discrimination based on height in the educational system, and taller people are promoted to, or admitted to college and so forth, that is captured in this educational PGS. I mean, like I said, there's been lots of analysis to find out what are the pathways, and most of them seem to be cognitive in nature, but there's no reason to suspect that other socially mediated pathways aren't at work here. So essentially we are, by using a polygenic score that sums across the entire genome, we're sacrificing the kind of molecular biology of understanding exactly the mechanisms by which the genes are having the effect on the downstream outcome for raw predictive power. And that's a trade-off I'm happy to discuss later. So how do we know this score is exogenous? If you look at this nice chart that looked at samples across Europe, if you extract the first two principal components of the DNA matrix, of the SNP matrix, you can see it predicts country of origin pretty darn well when you plot the first principal component against the second principal component. So you can see that the Irish, self-reported Irish, tend to cluster up in that corner of the 2D plot. The Spanish down here, even the Portuguese and the Spanish kind of overlap and separate out a bit. So one concern is that all we're doing with these DNA measures is getting another measure of different environments and different ancestries. So it's very important to control for these principal components. I'll show you the United States. This is our analysis of Ad Health, and you can see the first principal component along the horizontal axis tends to separate those of European descent and those of African descent, although you can see this is a a challenge, in a sense, to the social construction of race, is a challenge to our notions of race as a natural category, because there are white folks who are to the right on PC1 of African Americans who are to the left of those folks on PC1. You can see that Hispanics are a highly admixed population and combined of different ancestral groups, and Asians clustered down here. So if we didn't control for all these factors, then we would just be reproducing the effect of race. And I can only show 2 dimensions, 2 PCs on this plot, but typically we want to control for deep ancestry. So we'll control for 10 principal components. But we're more reassured by the fact in another study, a replication study we did in psychological science, that when you do compare the raw score, controlling for PCs, and then doing sibling fixed effects on the polygenic score, you see that they're pretty comparable. They don't change much. So what does that mean? When we do sibling fixed effects, like in Ad Health, for example, where we have full genome data and we have a sibling-based sample, we can compare 2 siblings who differ on their polygenic score, and really that's random assignment at birth, or at conception, really. So that is, by using the sibling comparison, we've broken any, with or without PCs control, which doesn't make really a difference, we have broken any potential linkage between tribal or ethnic or historical genetic ancestry and the associated environments with that, and the genetic difference that we're estimating off of, because they're full siblings, and it really is a random draw of the genetic lottery whether one sibling got 1 standard deviation above the mean in their polygenic score of education, and the other sibling was at the mean, let's say. And even though these show that they're pretty flat across the models, there are other analyses that I have that show that actually the effect is slightly bigger within families, suggesting that there's niche formation. So in other words, imagine a 1 standard deviation difference in your polygenic score between you and your sibling, you and a random other person, might result in an average. In fact, I'll show you what it results in. If I'm at the 3rd decile and Ian's at the 6th decile or 10th decile, I think Ian's at, across the population in the HRS, that predicts about a 1.5 years more schooling that Ian's going to get. So we found in some analysis that actually within families, that 1.5 year difference is actually bigger. So why am I emphasizing this? Because I think this is an important point of how we can have genetics matter, but through the familial, since the family accentuates genetic differences, it ends up in equilibrium, not resulting in genetic stratification across families when we reproduce the next generation. But I'll come back to that in the discussion. But you can see here, it predicts pretty well across deciles. I'll go back. It's normally distributed. There's concerns, and I'll get to those, about mortality bias and so forth in the HRS, given this is an older sample, but it looks pretty good, as you can see. So, for those of you not familiar with it, the HRS is a nationally representative longitudinal sample of older adults that started in 1992 with individuals age 50 or over and their spouses. And in 2006 and 2008, they collected samples that were later genotyped for the people who survived till 2006 or 2008, and then genotyped them for those 1.5 million markers that I talked about.
Well,
and
I know that you correct the mortality attrition. What about propensity to provide a sample?
Yeah, both of those.
You did both?
Yeah, I'll talk about that in the sensitivity analysis.
Groups are less likely
Yes,
to be
people who consent tend to be healthier and smarter.
Exactly. Well,
Okay, so, one last thing I want to say on this slide is that the entire analysis I'm going to show is going to focus on non-Hispanic whites. Now, That is because there are the discovery sample, the stuff that we got the betas from that created the PGS, was all from those of European descent, not African descent. And it's not a trivial issue because the greatest wellspring of genetic diversity is in sub-Saharan Africa and among the African diaspora. And therefore, what you're measuring is completely different. So you can't, there are apples and oranges. You could create a polygenic score within other races, but it's hard to apply them across races. And I can, again, talk about that. So we're going to put race, socially constructed race aside, or self-reported race aside for a moment.
So why are Hispanics a race? As a race?
Well, because they're admixed, as you saw from the so just to look at it before I get to the more controversial stuff, give you a couple examples to sort of what kind of analysis I'm talking about. So this is the genetics of smoking behavior. This is a polygenic score predicting ever smoked. Ever smoked is defined as having more than 100 cigarettes in your lifetime. Most people try, except for my class. They're so good. Most people try at least a cigarette or two. You're considered at one-time smoker if you smoked 100 cigarettes. Then it's all about the genetics of quitting. So you can see that over this birth cohort, the correlation between the polygenic score and smoking behavior has increased. This is not surprising. It replicates what's been found in twin studies. It replicates what's been found in other forms of analysis, that as the information about the ills of smoking became more widespread, the residual population that smokes is more hardwired, more genetically prone to not be able to quit. In fact, this helps explain the observation by economists that cigarette taxes, raising cigarette taxes, no longer has the same. Basically, the smoking elasticity is much lower now than it was 20, 30 years ago, because the residual pool of smokers is so biologically addicted to nicotine that you could raise a pack of cigarettes to $100, and they need it, right? So the genetics have kind of eclipsed environmental influences on smoking. To look at a couple other outcomes, and this is presented slightly differently. What would you expect in terms of height and body mass index? We've had a ballooning of the American population that's obviously environmentally induced from the 1980 Farm Bill and other factors. Would you expect a more environmental influence now than before? Well, essentially the budget constraint of calories and nutrition has been removed from the time of the Depression, or these early early cohorts. And you see now here, these two lines are someone who's at 1 standard deviation above the polygenic score for height and 1 standard deviation below the polygenic score for height. So comparing two parts of the distribution. And you see that this is significantly different, that the gap here is significantly different than it was in the early 20th century cohorts. In other words, the effects of genes are bigger now than they were back then, and they're much bigger for BMI. So this is a sort of cohort-based gene environment story where you remove the caloric constraints, and 1,000 flowers balloon, and sort of everyone can reach their genetic potential weight and height. So that leads us to the more controversial questions. The 3 propositions from the bell curve that I want to examine. So with in mind about how things change over time, we've seen 3 examples where a change in information ecology and a change in caloric ecology have all led to increasing genetic penetrance in those phenotypes over the 20th century. So it might seem reasonable that as schooling becomes more accessible to everybody, then we should see what Herrnstein and Murray predict: increased genetic stratification within schooling. So this is literally their text. High cognitive ability as of the 1990s means more than ever before the chances of success in life are good and getting better all the time, and these are decreasingly affected by the social environment, which by extension indicates that they must be increasingly affected by genes. So we can test that now that we have a measure of genetic endowment albeit a noisy one that has a lot of measurement error. I'll talk about that later. But what do we find? In contrast to the BMI and height story, and even the smoking story, we find that there's a significant decrease in the effect of genotype on educational attainment in more recent cohorts. Now, this is not the super recent cohorts, but this could be a story of the middle 20th century, and that for all of you, particularly students, that it's actually now widening again. We don't have data yet. We'll add help to probably if we do this. But it doesn't necessarily mean that when there's quote-unquote more opportunity, then you have increased genetic stratification. In this case, we see decrease. And I should add that this is not just the U.S. In the original Nature paper, we have a supplemental analysis that looks at this across Sweden, which you couldn't think of in terms of the West, a more opposite social policy, educational policy environment, and we see the exact same pattern, contrary to what our priors were. That expansion of educational opportunity in Sweden would have led to increased sorting by genotype, but instead it led to decreased sorting. Okay, the second proposition, that assortative mating is happening genetically, and this is going to lead to a widening of the distribution of talent or ability across generations. If you imagine this playing forward, this is exactly what chicken breeders do, right, to get maximal size and growth rates and all that, or cow breeders do for milk production. So is this happening to us in terms of education? Add to this phenomenon known as assortative mating. Likes attract when it comes to marriage, and intelligence is one of the most important of those likes. When this propensity to mate by IQ is combined with increasingly efficient educational and occupational stratification, assortative mating by IQ has more powerful effects in the next generation than it had in the previous one. This process, too, seems to be getting stronger in part of the brew creating the American class system. Okay, so this is an earlier paper we did, and we showed that there is indeed genetic assortative mating in the United States. One way to think of this is if it was random mating, there would be no shaded area. It would be on the 45-degree line. But we see a deviation from random mating such that this figure of the average pair of spouses is related at just under 5%. What does that mean? That means you're marrying your first cousin once removed, on average. I'll let you digest that for a second. If you control for ethnic background through PCs and everything like that, we can knock that down to second cousins. But on average, you're marrying somebody who's genetically similar to you as your second cousin. And this is the U.S., which has got no sort of history or tradition of first cousin marriage or arranged marriages and so forth. In Pakistan and the UAE, this is way higher. So where there's 60% of the marriages are first cousin marriages.
So can you just explain this to me better? What's the difference between at the bottom where there's more gray and the top? And don't understand the figure.
Think of it like a Gini with a Gini plot. Lorentz curve. Thank you. We're in a distribution that it's deviating from random. The important takeaway, I almost didn't show this slide. Now I'm regretting showing it for you. That to get into this is like a 3-year-old paper if you ask me. But basically, we created a relatedness, it's genome-wide restricted maximum likelihood. So basically, you go through you create a correlation across all SNPs among 26 million pairs of individuals in AHRS. And then you basically, the summative measure is the correlation in that relatedness score for spouses.
So what is this thing, the square at the bottom, what's that drawing my attention? The shaded area I see is 0.05, meaning that's the
deviation from random.
Okay, and it's narrower.
Yeah, but I'm not sure what that.
Okay.
I'd have to go back to the paper.
Okay, sorry.
But one question: this tells us, okay, we're marrying our cousins. That doesn't tell us that we're getting we're actually affecting the distribution of the genetics of educational attainment, or anything else for that matter, for the next generation, which would be Ernstine and Murray's claim. So if we look at that, we take the polygenic scores and we look at spousal assortment on those, the things that the genes that matter as opposed to overall genetic similarity. And we see that the lighter bars are the phenotypic correlations between spouses, and the darker bars are the genetic correlations. So you can see this huge, as we know, phenotypic correlation in education. It's not zero, but it's much less for the genotypes.
Is this the HRS data?
This is HRS, yeah.
So these are long ago.
We've done, I should say, we've done this where we do only first marriages, where we have marriages where one died before they got genotyped. We have ones where they're higher order marriages. We don't. This is pretty robust to all of that. BMI. In depression, there's no genetic correlation, even though there is one. This could be instead of, because we're measuring this late into their marriage, this could be actually reciprocal socialization. We had different genotypes, but since you're always eating, I'm going to eat with you. Or you're always depressed and you're bringing me down. I think the most interesting one is height, which seems to behave differently in a lot of the analyses, where the genotypic correlation is actually higher than the phenotypic correlation. So we're sorting on something that's correlated with height in terms of our endowment, but it's only partially height itself. So we know that height is like a summative developmental indicator of good child development. So that could be what's going on. But the real question is this changing over time, right? So we do this for these 4 phenotypes, and you see that in terms of, this is for the phenotypes, not the genotypes. There is, as we know, increased genetic correlation over time between spouses across the 20th century. That's not news to demographers. Even controlling for the marginals and all that. But when you look at the genotypic, nothing. Not for any of them. So we conclude that the increased sorting on the trend in sorting on education is not reflected by an underlying trend in genetically sorting on the genes that predict education. Okay, the third and final proposition I want to look at is that reproductive success is increasingly stratified by genotype. I'm really tempted to make an idiocracy joke now after the election, but I'll restrain myself. But you get the story that this has been a concern, dysgenics has been a concern for Lind and other right-wing psychologists for a long time, that basically because we observe a negative association between education and the number of children ever born since the demographic transition, we are making ourselves into a dumber population, despite the evidence about the Flynn effect. And there's lots of papers that try to reconcile those two phenomena. But again, now that we can open the black box and look at the genome, we can address this question more directly. I won't bother reading this quote. So when we look at same pattern, correlation with number ever born, overall in the sample, there's a negative correlation for education. There's a non-zero but much smaller correlation for the polygenic score. Again, height behaves really interestingly. Opposite sign for the phenotype and for the genotype, suggesting something about biological robustness or something. And the other two, no significant genotypic associations. That's BMI and depression, if you can't see the bottom. Again, the question, though, is the trend. Is it accelerating or is it decelerating? And again, we look at the phenotypes first. Education, much bigger effect. In fact, there was basically no difference by education among the first birth cohorts, and now there's a wide one. Nothing for height and depression, and for BMI, it's the opposite. Heavier people were penalized in terms of number of children ever born in the early part of the 20th century, and then there's no penalty in terms of your fertility for where you are on the BMI distribution today. But again, when we look by genotype, nothing. There's no significant interaction with birth year. So whatever's happening in terms of assortment in terms of association with fertility, it's happening at the environmental level, most likely. Okay, I mentioned there's a lot of issues here. Mortality bias. Luckily, we observed the initial sample, and we can develop weights to predict whether they survive to genotyping time and whether they agree to be genotyped. I think you asked that, Marta, right? Or Sarah. So we find that when we adjust for those weights, nothing changes. I mean, not nothing, but the story stays the same. I reran this on the Framingham heart study, the second generation sample, where the median age was 38 at genotyping, and so there's no significant mortality bias. We find the same effects. I don't want to front stage the Framingham because it's a bunch of white folks from Massachusetts. That is one of the more unrepresentative samples, even if you take race issues aside, if you just spoke, just on geographic basis and so forth. But at least it's reassuring that we're not seeing major differences. So another concern would be that there's a change in genetic architecture. So let's say that in my discovery samples, my discovery samples, the ones I said that the weights came from, those betas, the 1.5 million betas I got, that was genotype. Those were a bunch of folks that were born in the early 20th century. And then I'm applying those weights to people who are born early and late. They might predict better for the early because they're the same, whatever the genetic architecture for education is the same in the first half of the 20th century. And it's not that genetics have gotten less important in the second half of the 20th century, it's just that they've changed. What predicts educational success? Maybe you need more impulse control today and less cognitive ability, or vice versa. So it could be that we just have a measurement error problem across the cohorts. So my poor RA, I went back and made him get the age distribution for every cohort that went into all of these polygenic scores as best we could. We got about 70% coverage, and it seems like the age distribution matches pretty well, and if anything, the discovery sample was younger on average. The discovery sample were younger on average than the HRS, which suggests that any bias should be towards finding greater genetic effects later rather than in the earlier cohorts. So it might help explain smoking, BMI, and height, but it's not going to explain education. Finding an interaction effect with a very small marginal R squared. We don't have great power for a lot of them. I don't know if you can read this in the back, but for education, we're about 50% power. A little higher for depression, and height and BMI were pretty well powered. So that's a concern. That's largely due to the measurement error. As I mentioned, we think that there should be an R squared, if we measure things properly, of about 20% for education, based on doing relatedness matrix against a phenotype matrix on education. That itself is an underestimate of the total additive heritability for other reasons. But we use simulation extrapolation method, SIMEX, to correct for measurement error. I can talk about that in the Q&A, and our story doesn't really change. Another concern is that we're being confounded by changing overall patterns of marriage. So we know that in small towns, you're more likely to probably, at least throughout human history, maybe not in the United States, but in smaller communities, you're more likely to marry somebody who's genetically closer to you. And when you move to the city and we're in the urban marriage market, there's less descriptive sorting on marriage. So maybe we're just being confounded by overall genetic assorted mating changes. But when we look, we do find that in the older cohort, there's slightly higher. Remember that figure 0.045? We split the sample into two by the median age,
and we find it's slightly higher. They're not significantly different. When we look at number of ever born, the overall heritability of number of ever born is about 20%. It's pretty flat. I say sorry, R.A. Fisher, because he postulated that in equilibrium there should be no heritability of fertility, of fitness. Could only be a temporary deviation in evolutionary theory. However, a lot of people have observed is about 20% heritable. We looked at the urban-rural split. Doesn't explain what's going on. We also said, well, maybe people are just sorting more on the principal components, and that's another way to say that there's just overall sorting differences that are going on. So we split the principal components by older and younger and looked at correlations with spouses for the principal components, and we didn't find any differences. When we control for the principal components, there's no difference. When we don't control for them, there's no difference. It's all pretty robust. We take out spouses. We reorder the men and the women. We do all sorts of things to see if the way we're putting it in is driving things, and it's pretty robust. So I don't see a lot of evidence for what Herntling and Murray claimed, at least in trends. There are some small associations with fertility and with your spouse's genetic profile on these measures, but they're not increasing over time. So I'd like to move from Herntling and Murray in the last few minutes and talk about what I think is a more productive approach, sort of after hopefully slaying some, I don't know what you call it, beasts or whatever, to social genomics, and that is G by E, gene-environment interactions. If we just accept that, whether we like it or not, and I spent a lot of papers trying to knock down heritability estimates of genesis. I've challenged twin models, the Gremmel model I mentioned, and so forth, and I completely fail repeatedly to drive down heritability estimates. We know that for all social phenotypes, there's a moderate degree of heritability. It ranges from about 0.2 to 0.7. We just have to acknowledge that. So if we want to have a complete view of sort of the distribution of outcomes in society, wouldn't it be better to sort of integrate nature via nurture and gene-environment interaction effects? And the classic adage is to say that if there were such a thing as a genotype for aggression, and you grew up in the South Bronx in the 1970s, where Jimmy Carter's visiting, you end up in prison. And if you grew up in Mitt Romney's house, that's his house in Cranbrook, I think it's Michigan, you end up as Bain CEO, right? So how do we actually model that? So I'm going to argue that what social genomicists should be doing going forward is marrying the causal revolution in economics with these new genetic data to do G by E. So I'm going to give you one example of several today. This is the guy I call the dumbest politician. Again, that's a high bar today, but he was a congressman from upstate New York who thought even though 18-year-olds could not vote in 1969, their parents certainly could. But he thought it was good publicity to be the one saying next to Robert Moses, I think that is Robert Moses, pulling out the ping pong balls with the birth dates to send his constituents' sons possibly to fight and die in Southeast Asia. So this was the Vietnam draft lottery that replaced the deferment system that George Bush, Donald Trump, Bill Clinton, just the list goes on, all used to get out of the draft. This was supposed to be more fair. And what's really nice from a econometric perspective is that the last draft wave, 72, was never called up. So you could see the treatment effect of just getting a bad number, but never facing the prospect of being drafted. And Angus, in one of the first classic papers in 1990, looked at the effect of draft of veteran status in the Vietnam era on wages and found a 15% wage penalty in the 1980s for whites and 0% wage penalty for blacks. And the story was in a segmented labor market, if you're facing a poor labor market, regardless of your experience level, civilian experience level, it's not going to be a big hit for you if you're trapped cooking burgers if you're African American, but if you're white and you miss out on civilian labor market experience, it is going to have a hit on your productivity. That effect seems to have faded out by the 2000 census, but I'm going to show you that we're going to interact. We got the birth date data from the HRS, and we interacted the draft lottery status with, we do two-stage least squares, and we predict smoking behavior. So the troops were given cigarettes to help them stay alert and awake. The Army is a very tobacco-philic environment, even without that. So exposure, there is an average effect of being drafted on likelihood of smoking over the life course. And here we show that there's a significant G by E interaction, so that same smoking initiation genotype I showed before, you see that if you're one standard deviation or above on that score, and like a likely smoker genetically, and you're drafted, there's a treatment effect of, let's say, about a pack a day for the rest of your life. And this also translates to lung cancer risk. So here, I would argue, is a classic G. We have an exogenous shock to the environment. It's filtered through the individual differently based on their genotype and results in heterogeneous treatment effects downstream. So with that example in mind, this is why I think we should care about genetics within the social sciences. First, heritability and the overall sort of, when I keep referring to heritability, I haven't really defined it, but basically it's the proportion of the variation in a population of a phenotype that's explained by variation in the genotype. And we should care about that, contrary to what Arthur Goldberger, the late Wisconsin economist, once said in the 1970s, because he gave the example of the Royal Commission on Eyeglasses, and that we know that myopia is highly heritable, but does that change the treatment at all? No. We're not going to, if we find that the heritability of nearsightedness is 100%, doesn't mean that the Royal Commission on Eyeglasses should go pack up. But I think there's a second order question, though, is if we treat if we give you eyeglasses in your generation, are your kids still going to need eyeglasses? And if it's heritable, they are going to need eyeglasses still. And think about something like the culture versus genetic inheritance within families and the reproduction of poverty. I mentioned the interventions in Providence, Rhode Island, and elsewhere that are trying to increase the word count spoken by poor parents. If that's really the effect, it's environmental within the household, you do that one generation intervention, and it's going to have a ripple effect on the next generation. If it's genetic confounding, then it's not. You could still do that policy, but you're going to have to redo it every generation, essentially. So it tells us about the dynastic effects of our policies. We'll want given this as a confounder, by putting a polygenic score in a model, we can better control for the confounder and get more efficient and perhaps more consistent estimation of the variables we care about in social sciences. So imagine the Perry preschool study. You go back and you get their saliva today, and you genotype it, and you control for their educational polygenic score. You might find that the treatment effect of being getting the Perry preschool is actually bigger than we had estimated before, at least more precisely estimated. So there's another line of research that some folks are doing in epidemiology that uses genotypes as IVs to do Mendelian, what they call Mendelian randomization, to merely as an instrument to better estimate the effect of social variables. So, for example, you have a gene for alcohol dehydrogenase that predicts drinking alcohol. You instrument alcohol use with that, and then you can see whether or not the social effects of alcohol use on, say, wages. But there's huge issues with that about the exclusion restriction. There are some cases where I think it's a valid approach, particularly when you have a placebo test. But that's a developing area of research. As I mentioned with the Vietnam example, this can reveal heterogeneous treatment effects. Imagine you go to Baltimore, you do an intervention, or this reading intervention, or this word spoken intervention I mentioned in Providence, and you get a 10% treatment effect. You're like, ooh, I'm going to write this up in Nature. 10% is huge. But actually, you look at the data more closely, and you see that the vast majority gets zero effect in the treatment, pre-post. But for 10% of the kids, they doubled their reading scores. Why were they so responsive? Why are some kids devastated by family destruction or poverty or disorganization, crime in the neighborhood? And why are some kids dandelions? They're resilient. The intuition is that genotype is a prism that will refract the white light of average treatment effects into the heterogeneous treatment effects we can then see clearly. There's estimation issues about that, whether using polygenic scores are the right approach. I can talk about that one-on-one if you like. And then it can inform policy design, maybe. Imagine whole word versus phonics. And we could actually have an early indicator, a genetic indicator of risk for dyslexia or something like that. We could actually perhaps intervene earlier. And lastly, there's a consumer genomics revolution happening, whether we like it or not. It is something that social scientists should be aware of. Soon, 23andMe is going to meet OkCupid and is going to meet the fertility clinic. I thought about trying to apply for a patent, for instance, for the use of the polygenic score in dating apps, and in order to prevent it from being used by others, because I'm sure it's coming soon. And then it's going to end up in the fertility clinic. And already, as you saw, we have an R-squared for height that's now over 20%. So I want to end with showing you this clip. I don't know what was going on in the late 90s, but it was a very interesting time. In addition to the bell curve and Susan Mayer's book, the movie Gattaca, which at least my generation would be very familiar with. I don't know about the younger folks. Ethan Hawke, Uma Thurman, and I just want to play you this one scene from there. Maybe not. Well, I'll just describe what happened, which I really think is going to happen in the next 10 years, if not in the U.S., certainly in Singapore or somewhere else where there's less aversion, cultural aversion to this kind of thing, which is the guy, the geneticist says, it's still you, it's just the best of you, because they have IVF, they have a bunch of embryos, some are eliminated for like Huntington's and other like single gene diseases, and then they calculate the polygenic scores. They don't call it that in Gattaca, and they offer, well, you can have the taller kid or the smarter kid, etc. Of course, in the movie, I'll ruin it for you. Ethan Hawke, who's conceived in the back of a Chevy the old-fashioned way, ends up, outperforming his genetically limited potential. So I'll stop there and take questions. Thank you.